And good morning, everyone. So, exams are graded. I am going to post everything to Moodle and then hand out exams out during lab, but everybody did quite well, I think. So let's see, we got the little worksheet thing on cell signaling that is out there. It's a way of looking at some of the intricacies of cell signaling, and we're about to get into the last two sections of the class. First, today's and this week's discussion is going to be on components of the cytoskeleton, and then from there we'll be talking about the extracellular matrix and cancer next week, and have some things to discuss. Part of what we're doing is integrating a lot of things now that we've been talking about, and I think it's kind of fun. So I thought I would go through, just to finish up our cell signaling discussion, just to say we did, to look at a couple of the figures from the book to emphasize some of the critical points about how the different systems are working and how they can even work together, sort of self-signaling crosstalk, as it were. Wait for it. And so, as we've been saying, self-signaling is one of the truly critical parts of modern cell biology in terms of thinking how cells are able to know where they are, and cells are able to actually carry out differentiation. And one of the critical points is distinguishing between the different kinds of systems and actually beginning to think about how these different systems can, in fact, communicate within each other. And so we'll put some stuff up here. Another thing that's important, when we were talking about the plasma membrane and proteins in the membrane, there was a little experiment diagram we showed in which you can track a protein on the cell surface, and it turns out that some integral membrane proteins, if you recall, have sort of a wide path they can move. They actually have a fairly broad range of the cell they can move around in. And then there are other proteins that were very tightly linked. They didn't seem to move around much. Some did this, and then some did that. Part of this is things like G proteins and receptors, they have to stay pretty close together. There's a critical physical touching or connection between the receptor and its G protein, and the G protein and its effector. And so that portion of the membrane tends to actually hang together. And so there's actually a whole issue of what's called lipid rafts, which are contiguous parts of membrane that tend to kind of hang together. And that's because there are proteins that need to physically be near each other. And this kind of phenomenon we can imagine in the electron transport chain. You have to have all those components pretty much right next to each other. Additionally, with cell signaling that happens. And then there are other proteins there in the membrane, just as thinking about these, like different kinds of the sodium, potassium, ATPase, something like that. They actually can float around. Now, there is domains. I mean, the stuff that's on the apical side of the cell tends to stay on the apical side, and stuff that's on the basal side tends to stay on the basal side. And we'll look at some of those kind of connection changes and things in a bit. But some of these proteins do need to hang together fairly carefully. All right, so what we got then is these two broad cases of cell signaling, things that can happen that change protein function very fast, seconds to minutes. This is going to be essentially kinase cascades of some kind. And then we have things that are taking a very long time, hours, and this is going to be at the level of gene expression, altered protein synthesis. And so we know that steroid hormones are strictly operating at the level of gene expression. So we got these two broad classes, cell surface receptors, and they typically are going to be fast, and the intracellular receptors, things like steroid hormones, they can diffuse across the plasma membrane, and they're going to act at the level of gene expression. They're going to be pretty slow. Yep, here's a whole range of diffusible hormones for your dining and dancing pleasure. Go ahead and memorize them all. No, don't waste your time. But here's cortisol, and those of you that either work with Dr. Wilcoxon or etc. This is one of the stress hormones. And actually thinking about this, and it's one question I have to kind of ponder for a moment. He does an experiment where he will let people play some kind of like, Super Mario Kart, and look at cortisol levels. I guess that's what it is. Look at cortisol levels before and after. And cortisol is coming up because of stress. One of the things that's interesting is that stress response of cortisol is going to have a long-lasting effect, but it's not going to be very fast. It's not going to be like adrenaline. And so you have cortisol coming up because of stress, but the response to it is actually going to take a while. I'm going to think about that for a bit. And then, of course, there's the sex hormones, thyroid hormones. These are things that have inside cytoplasmic receptors. So this is a general diagram here. The activated receptor and hormone moves into the nucleus, and it now acts as a transcription factor. Yeah, let's roll. Let's see. Nitrous oxide, and there you go. All right, so now this is the broad range of things we can think about from a very cartoony perspective of intracellular signaling molecules and the things they can do. We have different kinds of enzymes that are present. They can be activated or inactivated. We are going to see there are a range of structural proteins, what we call cytoskeletal proteins. They're involved in cell shape change and cell movement. And then we're going to have things like CREB, which is the cyclic AMP responsive binding protein. This is a transcription factor that's waiting to be either phosphorylated, or it's waiting to have cyclic AMP bind to it. And it can then act as a transcription regulator, changing gene expression. And so this is going to be a long-term effect that is generated through this very rapid kind of response. And yeah, so here we are. Different kinds of things, amplification and all this stuff. A diagram about protein kinases, and a little diagram about GTP binding proteins. And so what the G protein is doing is it's in effect tickling this effector box. But good things to look at. So we have the ion channel receptor types. Those guys are certainly in the category of changing the cytoplasm very fast, but they're restricted to the nervous system primarily. And so what we're really focusing on are the G protein coupled receptors, or the enzyme coupled receptors. This is the term the book is using at the moment for what we refer to as the receptor tyrosine kinases. All right. And the G protein coupled receptors, the G protein is activated, and then in turn is going to interact with this little, I'm not sure what this is, some kind of stick. I don't know what you have sitting yourself. This is the effector that we said. We kind of generically talk about. The adenylyl cyclases are the phospholipase. Here's some foreign substances that act on cell surface receptors. And this is an interesting way to think about the fact that one of the reasons G protein coupled receptors are so well studied is they are the site of action of a ton of different drugs. So we got your basic downers, barbiturates, benzodiazepines, and these are interacting with GABA-activated ion channel coupled receptors, and they produce a feeling of relief, sedation. Nicotine, another interesting one, it is stimulating the acetylcholine-activated ion channel coupled receptors, that is, ligand-gated ion channels, in that it blocks acetylcholinesterase, and so you end up having the stimulation of acetylcholine on smooth muscle being carried out longer, so constriction of blood vessels, you have elevation of blood pressure, and all kinds of things happening in the brain. A couple of interesting guys, morphine and heroin. These are interacting with opiate receptors, which are G protein coupled receptors. And you have analgesia, relief of pain, that is, and euphoria, good. Curare, this is actually blocking the acetylcholine receptors, and it's actually the opposite of nicotine, and so you end up having muscle paralysis. Curare actually is still used in surgery. Often for anesthesiologists will actually administer a small amount of curare to paralyze some of the muscles so they can actually do the breathing for you, and you don't have, as the anesthesiologist, that pesky movement of the diaphragm and things. Let's see, a little bit of strychnine. Yep, good. And as we mentioned before, some of the interesting things with ligand-gated ion channels. We've got two compounds here you're familiar with perhaps, capsaicin from jalapeno peppers. This is stimulating the temperature-sensitive ion channel receptors to generate the sensation of heat, and menthol, which is generating the sensation of cold. So these guys, we have kind of a mix of ion channels and then some actual G-protein coupled receptors. I kind of like to, keep the ion channels story stuck to the nervous system, but still. All right, so here is kind of the structure of some classic receptors. One of the things you can do from a genomics perspective is that G-protein coupled receptors have a fairly standard structure to them, in that this part is somewhat generic in the way it passes around the membrane, and then there's going to be spots that are actually binding to the ligand that are going to be quite specific. So you can actually survey genomes and identify new G-protein coupled receptors. In fact, one of the things that's often referred to is an orphan receptor, in which somebody will find a receptor and not know what the ligand is, but the receptor is identified because it has this structure, and you can begin doing experimentation where you mutate it and see what happens. And the best thing is if you've got the receptor and the ligand as a scientific experiment, then you're really doing something. But very often, folks will identify a receptor and not have a ligand. Other people might be studying a ligand but not know the receptor. And let's see. All right, so here's a slightly more elaborate diagram of the G-protein coupled receptor system, in which the receptor is interacting with the G-protein. The guanosine diphosphate is released. It is so named because it binds GTP, and that actually causes the effector to be activated. So there is the subunit that is binding to the effector. The specifics of this are useful and worth keeping in mind that there's a lot to this, but not so critical to our overall discussion. We got the G-protein is activated here. And anything going on here? So G-proteins can actually change ion channels and things as well. It's a generalized kind of mechanism by which molecules are being generated within the cell. I kind of like my effector model better. Just as a diagram, here is ATP, and adenylyl cyclase is removing two phosphates and causing this cyclization, and you make cyclic AMP. Now, one of the interesting things, when we start talking about oncogenesis, is that we have to always have the tools by which we can silence these signals. So we've talked a little bit about kinases that add phosphates, and then phosphatases that remove phosphates. And it's easy to kind of focus on the kinases that are doing stuff, but just as critical are the phosphatases. In the same way, there is an enzyme that can take cyclic AMP and convert it into adenosine monophosphate, which, when we're generating energy, we pop a phosphate off and we generate adenosine diphosphate. But when you're incorporating nucleotides, you make adenosine monophosphate. Anyway, this is a molecule now that is not a particularly potent cell signal, and so you can quiet the system down by turning on cyclic AMP phosphodiesterase. So it's critical that you have systems in place that can turn the system on, but it's just as critical that you have systems in place that can turn the damn thing off. If we leave the system on, that is pretty much cancer, and we'll be seeing how that works. Here's a neuron that we add serotonin to, And in 20 seconds, we've got probably calcium being released or something dramatically happening here. Some different things that are mediated by cyclic AMP. Adrenaline, as we talked a little bit about, a major sort of fight or flight hormone, and that very quickly increases the heart rate and the force of the contraction. So you're pumping not just faster, but more blood. On the skeletal muscle, glycogen breakdown starts to generate all that different kinds of starch is being converted to glucose. Adrenaline is able to also initiate a long-term kind of food source by breaking down fats. And adrenocorticotropic hormone. This is actually one of the hormones that acts to cause secretion. And so it is secreting cortisol, and this is going to be then sort of a fast response that leads to a much slower long-term response. Let's see. Yeah, all right, so this is the breakdown of glycogen that we talked a little bit about, in that we've got the adenylyl cyclase is the effector. It activates protein kinase A, which requires cyclic AMP. Active protein kinase A is phosphorylating phosphorylase kinase. Phosphorylase kinase is phosphorylating glycogen phosphorylase. And finally, glycogen phosphorylase does something. It begins the breakdown of glycogen. So lots of different interesting pathways going on here. And additionally, as we were talking about, that we can have cyclic AMP engaging in long-term effects. In this case, we've got pKa making its way into the nucleus and phosphorylating a transcription factor, like Krebs. Let's see. All right, so now the other effector was phospholipase C, and a couple other guys, not quite as exciting, but still worth mentioning. Glycogen breakdown. Acetylcholine can respond in some tissues through phospholipase. Thrombin is involved in blood clot activation and stuff. And here is the diagram of the phospholipase activity. So the G protein tickles the effector. The effector is phospholipase. Phospholipase, in turn, is shaving off this part of the phospholipid bilayer. You generate an acetyl triphosphate, which in turn causes the release of calcium from the ER, and we also activate protein kinase C. And protein kinase C now is able to do a whole bunch of different kinds of activities as well. And let's see, this is actually an egg that has been fertilized at the arrow, and we see this wave of calcium come across the egg fairly quickly. The cam kinase, fine. Oh, let's see. Well, this is sort of an example of the kind of signal amplification that we can get. Not all that exciting. Okay, the receptor tyrosine kinase types. This is the alternative to a G protein coupled receptor, in that we've got the receptor tyrosine kinases. When they bind their hormone, they cross-phosphorylate. How do you do? They phosphorylate each other. You have these phosphate sites. These phosphate sites are in turn recruiting different kinds of signaling proteins that in turn can activate other kinases and all kinds of downstream pathways. And I will just tell you, that little diagram right there is a whole oncology class unto itself. So we'll be talking a little bit about cancer. One of the things that we'll be getting into is the idea of what are called oncogenes. And these are specific genes that, when mutated, are known to cause cancer. But one of the revelations, and we'll get into it next week, is the fact that oncogenes, these cancer-causing genes, in a normal, non-mutated form, are present in all cells all the time and in all organisms. And that wasn't a question, right? No. So you'll see things like RAS and FES and all kinds of different names. This is old-fashioned terms. The RAS protein is the cellular form of a mutated gene that's found in a virus known as the Rous avian sarcoma virus. So some of the names are a little odd, and we'll be getting into this. So one of the things that RAS can do is now turn on all these different kinds of kinases that are known as the MAP kinases. And so you can kind of imagine when activated RAS protein was first being studied, people came up and discovered this kinase. And it is referred to as the mitogen activated protein kinase. Mitogens are a generic term for anything that causes cells to divide, which in general is not a good thing to be sticking on your body. And so we have the MAP kinase. From there, further study realized that this was downstream, and it was downstream of the MAP kinase. And then lastly, the gene that was actually being activated by RAS is, of course, the MAP kinase. And blessedly, that's the top of it. I mean, we don't have a kinase. No. And this is having a whole range of effects. We got changes in protein activity real fast, and then we have changes in gene expression much slower. There's all kinds of things, and people will begin studying their very own kinase or their very own signaling protein. And, let's see. A couple of things you can do is carry out some interesting directed mutagenesis experiments and looking to see what the different kinds of phosphorylation spots on a receptor are doing. And so by doing this kind of careful work, you can begin to observe that if you change this spot to an alanine, that is not going to be a substrate for the kinase, because we're pretty much talking about tyrosine residues here, then it's not phosphorylated, and that changes the overall transmission of different kinds of proteins that are moving things into the next level of these different signaling molecules that are moving the signal forward. And this gets to be some fairly tedious genetics that people do, and there you go. Let's see. So, just some interesting things to think about. If you got a signaling system in which we have the signal molecule, and with a mutation in protein X, you don't get any signaling. You can introduce an overactive RAS. Essentially, it doesn't need to be triggered, and signaling is restored. So you would say then that protein X is acting upstream of RAS, because if you turn an activated RAS, everything starts to happen. In this case, if you've got a mutant and then you add the overactive RAS and you still don't get any signaling, that's a potential cancer treatment there, because it is inhibiting RAS activity. The protein is acting downstream of RAS, and so even though RAS is functioning, it's still not going to work. So we have lots of study that is involved in figuring out the order of the proteins in the signaling pathway. Anything else here? So this is actually a little bit of the diagram from the problem set. And what we got here is the notion that you can have a fair amount of crosstalk, which makes hormone cell signaling just that much more complicated. So you can have a hormone receptor system that is activating phospholipase that is then doing things. And then you can have a receptor tyrosine kinase that's also activating phospholipase in addition to other things. So it becomes an additive mechanism by which we can alter cell metabolism. And it is delightfully complicated. Let's see. Yep, there's all kinds of things that are working through this system. Good. All right. I think we got the PowerPoint on. What the heck? We'll look at the introduction to the next chapter. Chapter 17, the cytoskeleton. What we're doing here now is invoking the notion that rather than having a cell thought of as a balloon filled with enzymes, and the cytoplasm is in fact the density of probably really heavy jello. So rather than having a cell simply thought of as a water balloon filled with jello, which would be a very interesting kind of water balloon, there's actually a huge amount of structure within the cell. These are proteins that link together to form these different kinds of fibers and different sorts of structural elements underlying the plasma membrane and then stretching out to establish sort of tracks for our vesicles and things. And this is the realm of what's known as the cytoskeleton. And what we're going to do is work our way through the primary cytoskeletal elements. And here they are. Allow us to introduce to you your cytoskeletal elements. And you can make a little note of this, but we're going to take them one at a time. We have our intermediate filaments, microtubules, and actin filaments. And interestingly, the intermediate filaments have a bigger paragraph to them, but they're lamer. They don't do nearly as much stuff. And so we shall jump into it. And we're going to go back to the board now. All right. So again, we have a organization chart that you can put together. It's like a game for your response on buying proteins. Here I am raising my hands again. I assume that the YouTube has got some sort of AI crawl that looks through the video and finds something that's got some dynamics to it. I don't know. But it tends to have me as the thumbnail always doing that. So I'm going to have to figure out at the end of the semester how I can actually spoof the AI to do something that's going to be the thumbnail. I do want that. Come on now. I'll hang there. All right, we'll see. All right, so the cytoskeleton. And cytoskeletal elements are interesting things to study. For one thing, they tend to be really common in the cell. And so when we were talking about our lab experiment of our differential extraction, I made a notion that we'll be able to tell if we've broken apart a cell, because the cytoplasmic proteins have a very distinct kind of molecular weight signature. Specifically, actin and tubulin are in real high concentrations, and you always will see those when you crack open the cytoplasm. I mean, there's a ton of proteins in the cytoplasm, but in terms of an individual protein being really abundant, actin and tubulin are two major ones. Intermediate filaments are there, but they're not as easy to see, regardless. So we've got then these three classes of cytoskeletal elements. The intermediate filaments, and perhaps because they are less complicated, we take them first, but they were not the first to be discovered. Intermediate filaments, and then we've got microtubules, and lastly, microfilaments. And we got sort of a spiel to say about each one. And so what we're going to do is, rather than try and fit it all on one chart, we're going to take them one at a time. And let us start with intermediate filaments. So one of the things we'll see is that an aspect of these guys is how they polymerize. And one of the critical points about intermediate filaments is that there are a fairly broad class of proteins that act as intermediate filaments. And they've got similar kinds of structures, as we'll see in a bit. With actin and tubulin, they are a much more constricted gene set. There are multiple actin genes within the human genome and multiple tubulin genes, but with intermediate filaments, there's a whole bunch of them. And just a couple of the names of intermediate filaments. There's a protein class known as keratin, another one known as desmin, and another one known as lamin. This is underlying the nucleus. So these are just some of the names. If you see them thrown around, you go, oh, those are intermediate filaments. And one of the key points of intermediate filaments is they're going to line up like this, where we've got the amino terminus and the carboxy terminus. And they will auto-assemble into subunits that start to look like this, where we have the amino terminus and the carboxy terminus. The point is they form as dimers that then can begin linking together. And the critical point would be, if you flip this molecule around, it's the same. And so what we say with intermediate filaments is they have no polarity. And you might say, well, big deal, but I'll tell you, actin and microtubules, they have a very distinct polarity. Intermediate filaments are going to auto-self-assemble, and they form fairly tough structures, rigid structures. But pretty much all intermediate filaments are going to do is provide structural support. And this is essentially allowing the cells to have some toughness to them. I was looking over here, trying to keep the figures off so I can draw, just because I like drawing. But I'm missing them too fine. So intermediate filaments are made up of these big long ropes, and they've got these different kinds of classes. Let's see. But why not? It does a nice job. We'll throw it up. Stay. So here are intermediate filaments. We're looking at a fluorescent micrograph on the left side and then sort of a stylized drawing on the right side. There are these cell-cell connections, which we'll get to in a bit here, known as desmosomes. And, all right, this is an example of how we can begin building the intermediate filaments. And my diagram wasn't as exactly correct, but it's close enough. And then we have a coil-coil dimer where we have the amino terminus and the carboxy terminus. But then we're going to begin building these staggered guys where they've got the amino terminus in this direction, carboxy in that one, amino terminus this direction, carboxy terminus in that one. And so you build a molecule that has essentially no polarity to it. Either end is identical. These begin to build up into these really long, fairly tough filaments. And they are autocatalytically just forming up and making big filaments. So, some of the things these guys do. Just some different kinds of tissues and things. And these are primarily structural proteins. So just a little point. Keratin filaments are found in epithelial cells. So you can see here, I got some epithelial cells. And keratin is deposited in epithelial cells at a really high level. They're able to even further cross-link, and this is part of what gives your epithelial cells such tough structure. There are another class by Minton, which is in connective tissue. Neurofilaments are found in nerve cells, as the name implies. Nuclear lamins are found in all animal cells, underlying the nuclear envelope. And so this is an example of the effect of having a keratin mutation. And not all forms of keratin mutation generate this, but if you've got the coils such that they have mutations in the keratin protein, they can't form a nice coil. Whatever. The keratin protein is no longer structurally sound. And thinking about this is actually an example of a type of disease of epidermolysis bullosa, severe blistering that occurs. And it happens because the keratin is not really well tightly bound, and consequently it is not able to withstand the stress that our skin is subjected to. And let's see a couple other pictures here. And I'm not sure what this dude's trying to do. There's actually a class of defects in nuclear lamins that are very rare and cause premature aging disorders. But let's not look at that. That kind of freaks me out. All right. Back to the board. So our lamins are primarily generating structural support. If you are either missing or have specific defects, you can have different kinds of blistering in the skin. So we can recognize that structural capacity. Another thing that's happening is that if you take your skin and you stretch it like that, it's not at risk of being torn apart. Now, imagine if you take a piece of paper and you stretch it, depending like a piece of tissue paper, it pulls apart really easily. And there are tissues within the body, happily deep inside, you don't have to really worry about someone coming up to you and going, beep, and yanking them, that will begin to tear. Happily, our epithelium is really tough, and so it can actually withstand that kind of pressure and torsion. And this is in thanks to the intermediate filaments. Now, underlying the nucleus of every single cell is a class of proteins that are called the nuclear lamins. And this is an intermediate filament structure that is underneath the lipid bilayer nuclear envelope. Now, the nuclear envelope, in addition to being a bilayer, it's actually a double-walled thing. And so I think of it a little bit like a bunch of little sacs filled with fluid. These double walls actually begin to eventually integrate into the endoplasmic reticulum, and they are underlined with these intermediate filaments. This is what's providing the support for the nuclear envelope. Now, one of the first things that happens in mitosis, which you have perhaps covered in genetics, and it again is a subject of an entire another class, that is the cell cycle. And sadly, we don't get into the cell cycle as much as we should. But one of the first steps that happens when you know a cell is going into mitosis, a eukaryotic cell, is the loss of the nuclear envelope. So you have what's known as the S phase, where the DNA, the chromosomes are being replicated. And then a cell can hang there or not. It's going to then go into mitosis, which is the steps of prophase, metaphase, anaphase, telophase. And one of the things you know is that a cell is going into mitosis because you begin to see the chromosomes condense and you see the dissolution of the nuclear envelope. That dissolution occurs because there is, in fact, a kinase that is able to phosphorylate the nuclear lamins. It's actually a kinase known as the cell division cycle kinase, the CDC kinase, not the Center for Disease Control. The CDC kinase, and there's a number of them, they will phosphorylate nuclear lamins and cause them to no longer autocatalytically assemble, and this is responsible for dissolving the nuclear envelope. And so the nuclear envelope doesn't really dissolve, it just forms itself up into a bunch of little bubbles that can then reform. And so the nuclear lamins are a critical part of that as well. Let's see. So the nuclear envelope has the nuclear lamins, and there you go. So intermediate filaments are critical, but they're not doing a whole lot. What they're doing is providing structure. So they're kind of the rebar of a cell, in a sense. The next category of cytoskeletal elements is significantly more engaging in terms of doing stuff. And that is the class of cytoskeletal elements known as the microtubules. Microtubules are made up of two protein subunits, both called tubulin. And there is the alpha and the beta subunit of tubulin. So tubulin is a set of genes, but they are very, very similar genes. And they form initially first what's called a protofilament that is like this. And then the protofilament can begin to assemble into which you get essentially, it looks like a Twizzler. And if you don't know a Twizzler, well, that's your assignment. Twizzlers have a hole. A classic thing to do, of course, is to sip your soda through a Twizzler. You cannot do that with a piece of nerd rope because a nerd rope doesn't have a lumen, but a Twizzler does. And I don't know about red vines, but I do know at Twizzlers you have a hole through them. And so these protofilaments will autocatalyze themselves into a structure that looks like this. Where you have actually a little bit of a lumen. Now, the immediate question is, what's this filled with? And I think the best answer is a fairly stiff cheese-like material, very much like a combo. No, there is no evidence of anything actually specifically stuck into the middle of the microtubule. I actually heard a faculty member from the University of Texas ask about that. I'm like, no, dude, there is nothing in there. But it would be cool if it was a cheese filling. It's not. Now, the protofilaments, they actually can generate a variety of structures. You got your standard kind of tube, but they can also form something that'll look like this, sort of two C's that are linked around like that. A number of different kinds of structures, and people spend a huge amount of time counting the number of protofilaments. There's like 9 around them, and then there's different numbers. Really, do we care? I'm sure somebody does, but it's not anything that particularly excites me. So protofilaments line up, and they do bind a bit of nucleotide triphosphate, which is involved in their stability, but they don't have to have it. They're actually able to bind up without any kind of energy being placed into them. But they do have the GTP binding ability, but again, not all that critical. Now, one of the things we observe is microtubules have the ability to start out at a spot in a cell that is known as the microtubule organizing center. This is a spot where the microtubules will begin to coalesce and begin growing from. And a classic place to study microtubules is in nuclear or in chromosome division, looking at spindle fibers. And so, because you can identify a cell that's going into or chromosome division by observing the chromosomes lining up at the midline in metaphase, and spindle fibers are then growing out. And the spindle fibers are growing out from microtubule organizing centers. This is a spot where microtubules are essentially nucleating. Now, there are a number of microtubule organizing center spots in a cell. During cell division, or during chromosome division, this is known as the centrosome. And so we've got this spot in the two ends of the cell that are establishing as microtubule organizing centers, and from there the microtubules grow out, grab hold of the chromosome, and then they pull back. And one of the things this brings up is the fact that microtubules, MTs, they can grow and they can also shrink. And this is where we now have a difference between intermediate filaments and microtubules. Intermediate filaments, either end is the same. Intermediate filaments kind of build themselves up from both ends. Microtubules have got an end that is the real dynamic end, where things are being added and subtracted, and the more static end. And so we've got what is known in the phys, and it can be a bit confusing, but it is the minus end and the plus end. And truthfully, people have been studying this to understand how microtubules assemble. And so when you are looking at, say, an embryo and you fertilize it, or an egg, and it immediately is fertilized, the first thing that happens is this wave of calcium, and then you're going to have microtubules are coming out, and you begin to have cell division occurring. And it starts to happen very quickly. And this is a really good model system to study microtubule organizing. And so the spindle fiber production has become a classic way of looking at microtubules. And what we've got then is the plus end. And rather than thinking of it as the end that has the microtubules being added to it, it's also the end that has microtubules being removed from it. It is what I like to think of as the dynamic end. We have stuff being added and pulled away. And then there's the minus end. And this is a bit more static. So when we think about the microtubule organizing center in a dividing cell, that is a centrosome, we got the microtubules, spindle fibers, coming out like that. And the same thing happening over here. And they're going to meet up with the chromosomes and begin pulling them apart. And this is, in fact, the part of the cell cycle that we call anaphase, as the chromosomes are being pulled apart. And we will pick up on this on Wednesday. And that's all I have to say about that. So,
is the homework due Wednesday or Friday?
I would say the homework is due Wednesday or Friday.